Alright, hello everybody, my name is Gerard and I will be presenting the 1960s Thalidomide Catastrophe. My mentor is Lenny Tross, and this was a part of the Chemistry 211 honors course. In advance, I'd like to apologize for the Activate Windows watermark in the bottom right-hand corner of the screen, as I cannot get rid of that at the moment. Alright, so this research will be presenting the 1960s Thalidomide Catastrophe involving the creation, distribution, and recall of the thalidomide drug. Now, the drug itself was marketed as a sedative intended to ease morning sickness felt by pregnant women. However, unfortunately, the drug proved to be disastrous, causing one of the severest cases of birth defects seen in newborns today. Although the exact biological cause of these birth defects are still not fully understood, recent research has really furthered the link between the abnormal limb developments we've seen in newborns whose mothers took the drug, and the stereochemistry of the thalidomide molecule. So that will be discussed in the following slides. Now, my overall research goal here is to confirm the importance of stereochemistry in engineering new medications and furthering pharmaceutical research. Alright, so here's just a brief outline of what the research will be covering. So to begin with, in 1953 the drug was first synthesized by a Swiss company known as CIBA without any medical purpose, and it actually wouldn't be until 1956 that a West German company known as Grünenthal had got a hold of the drug. Now, at first they began experimenting with the drug in search of anti-epileptic properties, but instead they found that the drug had sedative properties. So after screening high dosages of the drug in mice, they were able to assume the safety of its consumption, and they found no LD50. Now, all an LD50 is a measurement of how poisonous a drug is. It's the amount of thalidomide that could be ingested before 50% of the sampled organisms, in this case the mice, had died. So with no LD50 established, the drug had successfully passed the regulation process at the time, which, as you can see, was pretty underdeveloped. So the drug was then distributed to throughout 46 different countries worldwide, but mostly in Europe, under different names, including thalidomide, Controgen, and Distaval. And as a side note, it actually wasn't distributed to the United States because Frances Kensley, a female FDA worker, had declined the drug's entrance in suspicion that it had caused neuropathy. So in 1961, as increasingly more use of the drug, more and more reported incidents of extreme teratogenesis, which is simply a hiccup in the limb development process, and mortality had been observed among physicians. So with up to 10,000 cases observed, obstetrician Dr. McBride and geneticist Dr. Lenz had began research on the link between these malformations in newborns and their mother's consumption of the sedative drug. And they were quickly able to establish a link between these malformations and the birth defects. So in 1961, the drug was immediately pulled from the market, first in England and then worldwide. So as a prerequisite to understanding stereochemistry, it's important to understand chirality. And all chirality is a qualitative property of objects. So a set of objects are considered to be chiral if and only if the two objects are mirror images of one another and they're non-superimposable. So looking at these set of hands here, we see that they are indeed mirror images of each other, and they cannot be superimposed. If one were to take the left hand and overlap it with the right hand, we would see that they wouldn't produce or form the same object. And now, while you might think rotating the left hand onto the right hand or vice versa would produce an overlap, they still wouldn't be considered the same object because they're facing different directions. Now, if we take a look at these flasks here, we can clearly see that they are achiral because while they are mirror images, the two flasks can be superimposed onto each other. So let's apply chirality to organic molecules. Now, all an organic molecule is a molecule with at least one carbon-hydrogen bond, contrary to the belief that it's a molecule that's naturally synthesized in an organism. So if we take a look at bromochlorofluoromethane here and dichlorofluoromethane, both of them would be considered organic molecules because they have at least one carbon-hydrogen bond. Now, the carbon atom is implied by the vertice here, because in organic chemistry we use vertices to imply the presence of a carbon atom rather than physically drawing it out. Now, all a stereoisomer is a set of chiral organic molecules. So if we take a look at bromochlorofluoromethane, the two versions shown would be considered stereoisomers because they are chiral. We can see that they are mirror images of each other, and they are non-superimposable even after a 180-degree rotation. So they would be considered stereoisomers. And they actually would be considered different molecules as well. So while they would both definitely be considered bromochlorofluoromethane, they are considered two different stereoisomers of bromochlorofluoromethane. Now if we take a look at dichlorofluoromethane, we can see that they are not stereoisomers because they are achiral. So if we take a look at the set here, we can see that while indeed they are mirror images of each other, they can be superimposed after a 180-degree rotation. So they are not stereoisomers. Now going back to stereoisomers, we can actually give each stereoisomer in a set a specific name. Now, using the system known as the Can-Angled Prelog naming system, what we do is we take a look at the order in which each atom surrounds the chiral center, and we rank their molecular weights. And by doing that, we can give each stereoisomer a title R and S. And when we have a set of stereoisomers which differ at one chiral center, we can actually call the set a set of enantiomers. So these two stereoisomers of bromochloromethane are considered enantiomers. We have the R enantiomer and the S enantiomer. So now let's take a look at the actual thalidomide molecule. So if we take a look here, we can see that we have the R enantiomer and the S enantiomer. So after further research was done, Dr. Lenz and other researchers discovered that thalidomide, specifically only one enantiomer of thalidomide, had been causing the severe malformations in the human embryo between approximately 20 to 36 days after fertilization. And that was the S enantiomer. So while the R enantiomer was effectively sedative and had the intended effect of easing morning illness in pregnant women, the S enantiomer had been causing the embryonic malformations. And for the first time in the pharmaceutical world ever, researchers had discovered that two isomers of the same drug can take drastically different biological pathways in the body and have different observed effects. So you might be wondering, well, if it was the S enantiomer that had been responsible for those malformations, and it was the R enantiomer that had been responsible for the intended sedative effect of thalidomide, then why hadn't the researchers simply isolated the R enantiomer and administered only the R enantiomer? And the reason they couldn't was because at physiological pH and the temperature provided by an organism's body, the R enantiomer can actually rapidly interconvert back and forth between its S and R configurations, so this makes even the consumption of the R enantiomer highly hazardous for pregnant women. So now let's examine how exactly the S enantiomer induces teratogenicity. So in general, thalidomide is an immunomodulatory drug, or an IMID, meaning it has a specific functional group that allows it to bind to the target, the cerebron protein. So this cerebron protein is abbreviated as CRBN and shown as dark green here. And what it does is when joined by a canonical substrate, simply a substrate that's naturally produced within the body, it will bind to this pocket here, known as the tri-TRP pocket. Now what will happen is it'll form this CRBN Col4 complex, which is simply the name we give for the arrangement of proteins formed once this substrate will bind. And once joined by ubiquitin factors, which are represented by these orange circles here, the E2 proteasome is actually going to degrade this substrate and recycle its materials. So this allows the cell to conserve energy and conserve precious materials when a particular substrate isn't being used in the cell at a specific time. So what's important about this process here is that cell 4 will not be affected by the binding of the substrate to the CRBN protein. Because the substrate takes the place of this binding site, cell 4 will not take the place of the binding site, and it'll simply continue along and regulate gene transcription. Now, as for what it specifically regulates, cell 4 will serve as a regulator of gene transcription for specific key transcription factors that are needed for embryonic development. So in other words, cell 4 will be needed to produce those key proteins for limb development. So in the presence of an immunomodulatory drug, however, we see that thalidomide will be binding to the CRBN protein, and specifically S enantiomer will bind to it. Now, while the R enantiomer will still technically bind to the protein, as confirmed by a study in 2018 with zebrafish cells, the S enantiomer will bind with an affinity that's 10 times stronger than the R enantiomer. And when that happens, it's going to change the CRBN conformation or structure in such a drastic way that cell 4 is now going to be able to join this complex. And when we have cell 4 joining the complex and the ubiquitin factors subsequently joining, the E2 proteasome is now going to degrade that cell 4 protein. And as a result, we won't have that essential regulator of gene transcription, so those key proteins needed for limb development won't be produced. And in turn, we will observe teratogenicity. So going back to this binding here, while only the S enantiomer will induce this biological pathway, the R enantiomer can once again intraconvert into the S enantiomer and induce the pathway. Now, this means that this once again confirms that consumption of either enantiomer or even a racemic mixture of thalidomide is highly hazardous for pregnant women. So now let's take a brief look at how exactly thalidomide is synthesized today. So after experimentation with new reagents, pharmaceutical companies have been able to optimize the original synthetic pathway used by Grunenthal to manufacture thalidomide. So in general, in organic chemistry, a synthetic pathway, such as the one shown here, is simply a stepwise procedure showing what reagents or chemicals and solvents we need to add into a flask of our starting material to create our target molecule. Now in this case, our starting material is phthalic anhydride, and our target molecule is of course thalidomide. So often a synthesis will include some intermediates because there isn't simply one reagent that we can add to our starting material to directly convert into our target molecule. And in general, the less intermediates we have, the better, because after each intermediate step in the synthetic pathway, we're going to lose some of our yield due to natural error. So looking at this synthetic pathway, because only one intermediate is used, it's going to have a relatively high yield, and we could see that as after multiple trials, we'll obtain a maximum yield of about 56%. Now, in addition to this relatively high yield, this synthesis starts with phthalic anhydride, which is relatively inexpensive, about 20 cents per gram, depending on the supplier, the packing, its purity, and some other factors. Now, on top of that, we also have the fact that this synthesis doesn't use many. In fact, it uses no toxic solvents. So we have pyridine here, and we have ammonium acetate as our solvents for steps A and B respectively, and these are relatively non-toxic. So running this reaction on a higher scale won't be as environmentally damaging as the Grunenthal synthesis. So how exactly is thalidomide being used today? Well, thalidomide has actually found multiple uses in the medical field, despite its teratogenetic effects. So having 4 acyl groups, or 4 C double bonded O groups, as seen here, and having 2 amide groups, this allows thalidomide to have a diverse range of biochemical pathways as seen in the photo. And as a stimulator of the immune system, for example, thalidomide is considered to be an immunomodulatory, which means it promotes T-cell activation and the rupturing of cancer cells. In addition, thalidomide has found its way in treatment of multiple myeloma. Now, multiple myeloma is a form of cancer in plasma cells. Plasma cells are a type of white blood cell found in bone marrow, which once cancerous can release abnormal antibodies and kidney damage, and thereby weaken the immune system. So, in addition, we also see that thalidomide is anti-angiogenic and anti-inflammatory, and this allows it to reduce tumor growth in blood vessels, which is a key mechanism in one of its anti-cancer roles. And as with any drug, of course, consumption of thalidomide will come with side effects, such as drowsiness, since it's again a sedative, blood clotting, rash, and in some cases a form of nerve damage, known as peripheral neuropathy. Now, currently researchers are experimenting with other similarly structured drugs, which are known as analogs. So these include lenalidomide and polidomide. These differ from thalidomide by a single element. However, after some research, they've actually found that these drugs in comparison to thalidomide have reduced side effects and have a stronger ability to inhibit or stimulate certain proteins in whatever biochemical pathway they're analyzing here. Alright, so here are my works cited. and thank you for your participation.